going to look at a brief overview of the process, including the MCMC, Markov chain Monte Carlo portion, and we're going to emphasize the strength of PG to provide exclusions and to get more information from the data files. Master software will be used as an example of a hypothesis testing, including some cases with assumed contributors, analyses with multiple persons of interest, and deconvolution of a mixture to provide investigative leads when there are no assumed contributors or persons of interest available. Here are some more specific topics. We'll look at the SWGDAM guidelines, and we're going to start here because I think most people are familiar with the guidelines and very familiar with the characteristics of the electropherograms. Then we'll move on to looking at examples of variation and complexity that will often cause difficulty with the analysis. And how's modeling used in continuous probabilistic genotyping? And we'll specifically take some time to look at system parameter data sets. In section 4, we'll have a presentation on validation of PG software. And in section 5, we'll have examples of PG software workflow and results. So a summary of the PG criteria. All of these are taken from the SWGDAM guidelines. And just in general, these are an overview that this is biological modeling. They're looking at probability distributions to calculate likelihood ratios and or infer genotypes. And it's a tool very important to assist the analyst in interpretation. It's not intended to replace human evaluation. It's particularly useful for low-level DNA samples and complex mixtures. And the probabilistic genotyping approaches can reduce subjectivities in the analysis of DNA typing results because we're using the information from the traces and applying rules across the board. Historical methods of mixture interpretation consider all interpretive genotype combinations to be equally probable, whereas the probabilistic approach provides statistical weighting to different genotype combinations. So weighted genotypes, and it does not include a stochastic threshold. Instead, it's incorporating the probabilities of alleles dropping in or out, and it uses more of the genotype information, thus enabling the ability to distinguish between true contributors and non-contributors, and especially in situations where potentially via earlier methods, the conclusion would have been cannot exclude. Here you have using the weighted genotypes, you have a likelihood of ratio that can be very negative and give weight to exclusion. It may require prerequisite studies to establish parameters such as stutter expectations, peak height variation, and so on. So this is a summary of PG criteria that prerequisite studies, and here's just an example of high peak with nice balance. And we get lower peak going from thousands to hundreds. We're getting some peak height variation, even very close to each other. A little bit of maybe PCR bias here with the smaller fragment. It's not in a stutter position, and it's 3 base pairs, not 4 away. And then at the hundreds here, we do have extreme variation. And so the prerequisite studies, this is just an example of peak height variation that's needed by the software to be able to appropriately model the mixtures. Okay, here's another example showing dropout. So these are much higher RFU peaks, and we have a nice balance. This is the same sample, but at a lower RFU, and you see that it reached the dropout, and it is actually flagged here for being above analytical but below stochastic. Indeed, that there is a dropout peak there that's below analytical threshold. So this is an example of some of the information that is obtained and provided to PG software to give it context for evaluating the mixture samples that you provide at a later time. Okay, so what would like to look at is look at examples of variation in complexity that cause difficulty with analysis. These will be very familiar, as I'm sure most of you have had samples like these, but it's a nice starting point. And then we'll move into how modeling is used. Okay, so examples of variation in complexity, just the genotype sharing of combinations. So a two-person mixture might have 4 peaks, might have 3 peaks, might have 2 peaks, or might have a single homozygous where everything's overlapping and they're both homozygous. So variation in a two-person mixture of some examples of allele sharing and peak height variation. If we go to a 3-contributor mixture, now we've gone to 150 possible allele combinations. And a 4-person mixture has over 600 possible allele combinations. So it becomes very evident that an individual person would have extreme difficulty in considering the different allele combinations, stacking, peak height variations, stutter, drop out, drop in, at these mixtures that are 3-person or higher. Here's a nice image taken from John Butler's textbook, where if the observed data is 3 alleles and there are varying peak heights, what's the actual composition? And so it's showing that you've got 3 minor contributors and 1 that's slightly higher. And so we've got one individual that's an 11-2, I'm sorry, an 11-12. And then we have a minor contributor at 10-10, homozygous, another minor contributor that's 10-12, and another minor contributor that's 10-11. So when first looking at this, it would be difficult to determine this composition used from the stacked alleles. Another source of variation is the chemistry and the procedures used, 28 cycles and 1 unit of TAC, compared to 32 cycles and 2 units of TAC. And so here, with the fewer cycles and 1 unit of TAC, you start to see peak height variation that's very noticeable at 20 picograms, and with greater cycles and more TAC, and a little less variation. At 10 picograms, we're starting to get dropout in the fewer cycles and less TAC. Not so much of an impact here, again, at the 32 cycles and more TAC. And when we get down to 5 picograms, that's when we start to see the dropout here for the 32 cycles and 2 units of TAC. So the chemistry in the SOP will make a difference in variation. Also, degradation of the signal. Okay, so larger multiplexes have loci with higher molecular weight, less template is available for PCR, and so you just have this normal degradation of signal with the higher molecular weight peaks. And then there's also, of course, degradation of DNA, depending on the quality of the DNA. Okay, another source of peak height imbalance, looking at heterozygous balance typically decreases with the template level of the DNA. And we saw that 2 slides previously. And this is just another example going all the way down to 0.05 nanograms. Where, here at this range of concentration, we're starting to see peak height variation, 60%, 40%, all the way down to dropout of the allele. Okay. So that was just a summary of things to remind us that there are a lot of reasons that there's variation in a trace. Okay, so modeling with continuous PG analysis is an excellent helpful tool. And what the model is doing is it's looking at characteristics like stutter, peak height variation, degradation, allele sharing, and it looks at the genotype that are in the sample and then presents different models that would explain those peaks, what would be expected given a certain model. And here's a note saying the lab will generate known samples, replicates, and dilutions that are required by models to provide an accurate picture of expected peak heights, which was also noted in the Swigdam description. Okay, so why is it complicated? Well, we're looking at thousands and thousands of different iterations. Remember, there were over 600 combinations just of the allele cults when you were looking at 3 or 4 person mixtures. Then you get up to 5-person mixtures, of course, even more. So a very large number of interrelated model parameters. So it's the probability of the data given the model is equal to the integration of all of the different parameters. MCMC can approximate this with very high accuracy, and M represents all the various model parameters. Okay. All right, and I think most people are familiar now with Markov chain Monte Carlo approach to assess likelihood that a suspect is a contributor based on the evidence. And this photo credit from University of Connecticut is describing it in very simple terms of a collection of potential parameters, and then look at another set of potential parameters, compare it. Is it closer to the actual that's in the sample? And then go to the next set and so on. And this repeats over several iterations until it comes to a convergence and that it has weighted the most likely genotype combinations that would explain the data. Okay. So a few years back, I was at a presentation by Mike Coble, and this combined with the reference from Taylor, Bright, and Buckleton is very helpful, I think, in visualizing these model parameters for each iteration of MCMC. And so using just a 2-contributor mixture and 2 loci for the illustration, we're going to look at the different parameters for each iteration. So one parameter is the genotype. What are the allele calls? So in this small example, we have one locus that has 2 peaks and another locus that has 3 peaks. Okay, so the allele combination is one parameter. Another parameter is the zygosity. If contributor 1 is heterozygous for 15 and 16, then you would have approximately equal height peaks at 15 and 16. And if it's homozygous for the second locus, we would expect to see this 5 allele significantly taller. Okay, and if contributor 2 is homozygous for 16, that 16 is going to be a taller peak because it has 1 allele from contributor 1 and 2 alleles from contributor 2. If it's heterozygous for 6 and 7, then we would expect those to be approximately the same height. So 2 alleles from contributor 1, but they're stacked, or they're homozygous, and 2 alleles for contributor 2 heterozygous. And when we redraw it, now the electropherogram, instead of having 5 evenly height peaks, has this pattern. So that's considering zygosity. Also the template amount. If contributor 1 is a major contributor, 15, 16, and 5 are going to be slightly higher. So 15, 16, and 5 because it's a major contributor. If contributor 2 is a minor contributor, that's 2 alleles slightly lower, and these 2 also lower because they're from contributor 2. And so then we redraw it, and these have become even shorter, and this one has become taller. Okay. Another is degradation, the available template. So if contributor 1, member is a major contributor, has more DNA, going to have slower degradation. Typically, if you start with less DNA, you're going to have less due to the degradation. And so now the peaks that were associated with contributor 1 have degraded less and or have a lower RFU, and the peaks from contributor 2 because of degradation are lower. Okay, locus-specific amplification is another potential. And if locus 1 is above average, those might be slightly higher. And if locus 2 is average, no change. Okay, so it's a little slightly higher on both of those if there's locus-specific amplification efficiency. And of course, stutter. So we're looking here at the main peaks and potential stutter peaks. So 15 could have a stutter in the 14 position. 16 might have plus stutter. 15 is also getting stutter off of 16. So even though it's an actual contributor peak, some of this will be from stutter. And so then we see the same thing here. 5 is the peak, might make a stutter peak. 6 and 7, lower RFU. They do contribute to 5, but less noticeably. Okay, so at the end of the iteration, what the model is, it's comparing the model profile to the data. And so I have the model drawn in purple and the actual data in green. And so here we're taking a look to see if they're similar, it's accepted, and this is the new start point. If they're not similar, it's rejected and it goes back to the that original one and then picks another set of parameters to model. Okay. Modeling and continuous PG analysis uses Metropolis-Hastings equation for the Markov chain Monte Carlo. And this is incrementally moving from the sampling of an area of higher to an area of higher probability. And it's still sampling the tails of the distribution. Burn-in is a term that's used to describe the early sampling, tails, for the MCMC. And there are different MCMC robots online that are helpful for visualizing this. Basically, it is an incremental move. Does the point chosen at random come closer to the data trend? Then pick another one. Is it similar or different compared to the data? If it's closer, you accept it, and this is the new sampling point. If it's further away, you reject it, you keep the current point, and you sample again. Okay, so the next thing we want to do is explain the system parameter data set. This is very similar to the calibration data set that you heard about for Knockit. And this is a diagram to help visualize the system parameters. It's calculating a variance factor. The variance factor is 1, it's low, it's quite tight. And as you get down, the expected peak height is on the x-axis ratio, and the log ratio of the observed over expected is on the y-axis. So at higher peak heights, you expect to see nice balance between a heterozygous individual at a locus. As you get down in peak height, you're not surprised that this is still a single person, just heterozygous. This is also from one single source file, but it's very low, and it has an RFU here of the tallest peak only at 250. And you're not surprised at that low RFU that there's a very significant, it's less than 50%, the peak height balance. Okay. So these are all examples of the samples that you provide to the software as part of the system parameter estimation or calculation. You're using dilutions and replicates to show the peak height variation over the range of DNA concentrations. You're wanting to select samples that have a wide range of peak allele calls so that you can get capture as much stutter information and as much variation because of size as possible. And pointing out that the system parameters are unique to the chemistry, the instrument, and the SOP. So if the chemistry or the instrument or the process used to produce the FSA files changes, then you would want to look at your system parameter files again to make sure that things are still accurately giving a picture to the software of what to expect in terms of variation in single source samples. Okay, so here's just an example of the file template. We've got the samples on the image on the left, and this is just the allele calls and their peak heights. So this is giving the information of what's typical peak height. And then these samples are also analyzed, including the stutter. And so here we've got all the allele calls. As you can see that it was, it was a 17-18 with a 16 stutter, and 12-13 with the 11 stutter.